Assalamu alaikum. Today we will talk about intracellular receptors. Subsignal molecules are hydrophobic and are small enough to pass rapidly across the targeted cell's membrane. Once inside, they directly regulate the activity of specific intracellular proteins. Functions are receive signals from lipid-soluble or non-charged, non-polar small molecules, trigger a variety of responses in the cells depending upon the function of the receptor. Intracellular receptor structure they have no extracellular signal binding site, location, cytoplasm, nucleus, endoplasm reticulum, intracellular residues. Examples are gaseous signal molecules that bind to intracellular receptors are nitric oxide or carbon oxide. Non-gaseous signal molecules that bind to intracellular receptors are steroid hormones, vitamin D, thyroid hormone, returnic acid, etc. The gaseous signal molecules that bind to intracellular receptors. One of its example is nitric oxide. Let's discuss nitric oxide in detail. Nitric oxide is highly reactive gas. It acts as a signal molecule both in animals and plants. Even some bacteria can detect very low concentration of NO and move away from it. Synthesis of nitric oxide. It is made by the deamination of the amino acid arginine catalyzed by enzymes called nitric oxide synthase. Nitric oxide synthase converts arginine into a metabolic citrulline and simultaneously generates nitric oxide, as you can see in the picture. Genes that encode nitric oxide synthase in the human and the mouse genome contains three different genes that encode nitric oxide synthase. Nitric oxide synthase NOS1 found in neurons, hence the N and muscle cells. INOS found in macrophages. The I stands for inducible, whereas the levels of NOS and ENOS are relatively steady. Expression of INOS genes awaits an appropriate stimulus, that is, ingestion of a parasite, called inducible NOS because they made it only when they are activated, usually in response to an infection. ENOS, NOS3 found in the endothelial, hence the E, cells that line the lumen of blood vessels. All type of NOS produce nitric oxide from arginine with the aid of molecular oxygen and NADPH. Cell signaling type. Nitric oxide diffuses rapidly out of cell where it is produced and into the neighboring cells. As it is quickly consumed close to where it is synthesized, so nitric oxide acts in a paracrine or even autocrine fashion, affecting only cells near its point of synthesis. It acts only locally because it has a short half-life, about 5 to 10 seconds, in the extracellular space before oxygen and water convert it into nitrates and nitrites. Mechanism of nitric oxide action. The signaling function of nitric oxide begins when it binds to protein receptors or in the cell. The binding sites can be either a metal line in the protein or one of its S atoms, for example orange cysteine. In either cases, binding triggers an allostatic change in the protein, which in turn triggers the formation of a second messenger within this. Blood flow. In mammals, one of nitric oxide function is to relax smooth muscles in the walls of blood vessels. Autonomic nerves in the vessel's wall release acetylcholine. The acetylcholine acts on the nearby endothelial cells that line the interior of the vessel, and the endothelial cells respond by releasing nitric oxide, which relaxes the smooth muscle cells in the wall, allowing the vessel to dilate. Nitroglycerin has been used for about 100 years to treat patients with angina, pain resulting from inadequate blood flow to the heart muscles. The reason is nitroglycerin is converted to nitric oxide, which relaxes blood vessels. This reduces the workload on the heart and as a consequence reduces the oxygen requirement of the heart attack. Blood flow. Kidney function. Releases of nitric oxide around the glomeruli of the kidneys increases blood flow through them, thus increasing the rate of filtration and urine formation. Action on smooth muscle. Peristalsis, the wave-like motion of the gastrointestinal tract, are aided by the relaxing effect of nitric oxide on the smooth muscles in its walls. Birth, nitric oxide also inhibits the contractility of the smooth muscles wall of the uterus. As the moment of birth approaches, the production of nitric oxide decreases. Nitroglycerin has helped some women who were at the risk of giving birth prematurely to carry their baby to the full term. In some target cells, including smooth muscle cells, nitric oxide reversibly binds to iron in the active site of the enzyme guanylyl cyclase, stimulating this enzyme to produce the small intracellular signaling molecule cyclic guanosine monophosphate. Thus, guanylyl cyclase acts both as an intracellular receptor for nitric oxide as an intracellular signaling protein. Most of the effects of CGMP are mediated by CGMP-dependent protein kinases, also known as protein kinases G. In vascular smooth muscles, PKG activates a signaling pathway that results in inhibition of the actin-myosin complex, relaxation of the cell dilation of the blood vessels. As you can see in this diagram clearly, the NO produced by NOS3 inhibits inflammation in blood vessels. It does this by blocking the exocytosis of mediators of inflammation from the endothelial cells. Effects upon secretions. Nitric oxide affects secretion for several endocrine glands. For example, it stimulates the release of gonadotropin release hormone from hypothalamus, the release of pancreatic amylase from the exocrine portion of the pancreas, the release of adrenaline from the adrenal medulla. Killing pathogens. Nitric oxide aids in the killing of engulfed pathogens like bacteria within the lysosomes of macrophages. Plants also use nitric oxide. Nitric oxide is involved in the defensive response to injury or infection. Carbon monoxide is another example of gaseous intracellular receptors, another gas that is used as an extracellular signal molecule and like nitric oxide can act by stimulating guanylyl cyclase. Some known gaseous signal molecules that bind to the intracellular receptors are various small hydrophobic signal molecules diffuse directly across the plasma membrane of targeted cells and bind to intracellular receptors that are gene regulatory proteins. Examples of these signal molecules include steroid hormones, thyroid hormones, vitamin D, and retinoids. Cortisol produced in the cortex of adrenal influences the metabolism of many types of cells. Steroid sex hormones made in the testes and ovaries responsible for the secondary sex characteristics that distinguish males from females. Vitamin D synthesized in the skin in response to sunlight. It regulates calcium metabolism, promoting calcium 2 plus uptake in the gut and reducing its secretion in the kidneys. Thyroid hormones made from the amino acid tyrosine act to increase the metabolic rate of many cell types. Retinoids such as retinoic acid made from vitamin A have important role as local mediators in vertebrate development. The structures of some are there, you can see them clearly in detail. Nitric oxide is highly reactive gas. It acts as a signal molecule both in animals and plants. Even some bacteria can detect very low concentration of NO and move away from it. Some known gaseous signal molecules that bind to the intracellular For more videos, subscribe to Subject Expert and please like and share the video. Thank you.